The human body is made up of around 200 different types of cells, nearly all of which have the same DNA, but only certain regions of DNA are expressed within different cell types. How DNA is expressed is determined by a multitude of factors, and an error at any stage of expression could result in an incorrectly functioning protein. For example, people with hypothetical disease X have improperly functioning liver cells, so their livers are unable to break down substances properly. Disease X is caused by a mutation in gene B. What errors in DNA expression may have caused the mutation in gene B? Within the nucleus of each cell, DNA is wrapped around spool-like structures called histones as a means of storing and condensing DNA. How tightly DNA is wrapped around a histone is a major factor in whether the DNA is transcribed because, if wrapped tightly, the DNA is inaccessible and unable to be translated. But if the DNA is loosely wrapped, it is accessible to transcription enzymes and can be transcribed into pre-mRNA. Here are the basic mechanisms of histone modifications. A positively charged histone binds with the negatively charged phosphate backbone of DNA, allowing the DNA to wrap around the histone protein. DNA that does not need to be transcribed is wrapped tightly around a histone protein. This tightening is induced by methyltransferase, an enzyme that adds methyl groups to the DNA, making it inaccessible to transcription enzymes. DNA that is ready to be transcribed is loosened by the addition of an acetyl group, an action performed by the enzyme acetyltransferase. The acetyl group neutralizes the negative phosphate groups of DNA, weakening its bond to the positively charged histone protein. The addition of acetyl groups by acetyltransferase and the removal of methyl groups allows transcription enzymes to access the DNA and carry out transcription of the gene. It is possible that gene B is not properly expressed in the liver due to overmethylation of DNA, preventing the DNA from being accessed and transcribed. Once pre-mRNA has been formed via transcription of DNA, it must undergo RNA processing to form functional mRNA, which is then transported out of the nucleus to be used for protein formation. RNA processing includes the removal of introns or non-coding regions of the pre-mRNA so that only the exons or coding regions are left. These necessary exons form the functional mRNA that will exit the nucleus and undergo translation into a functional protein that will break down substances within the liver. As with any other biological process, errors may arise during the splicing of a gene. For example, a splicing factor may bind to the exons necessary for the formation of gene B in pre-mRNA, resulting in them being spliced out along with the introns, leaving an incomplete and non-functional mRNA. This non-functional mRNA cannot be translated into the correct protein, potentially being the mutation that prevents the gene B protein from being produced and, therefore, the reason liver cells are unable to properly break down substances in individuals with hypothetical disease X.